Hello friends, welcome back to my YouTube channel Astin Padasalai. So in this video, I'm going to tell you top 100 important topic that you need to prepare with regarding to this BARC Biosciences 2024 examination. So without any delay, let's get into the video. First topic is gene transfer methods in plants and animal. With special focus you need to give for agrobacterium mediated gene transfer. So in 2023, there was a question asked from agrobacterium mediated gene transfer. You need to know about the basics followed by that where genes and their function and also the TI plasmid and recent innovations like you need to know about binary vector system, co-integrate vector system, and also you need to know about the gene transfer methods in animal. Next is cancer biology. So cancer biology is a very important topic. Sometimes they will be asking you direct questions. Sometimes they will be an application like CSR part C kind of questions you can expect in BARC examination. Last time there were two questions came from cancer and again two questions came from apoptosis. Okay. So cancer again you need to know the basics and also you need to correlate the cell cycle and cancer. Next the therapeutics of cancer. Okay. And also you need to study about the mRNAs involved in cancers. Okay, followed by that apoptosis. Both you need to know about the intrinsic and extrinsic pathway and list of pro-apoptotic gene and anti-apoptotic gene and all those things you need to cover up. And also you need to know about the difference between apoptosis and necrosis. What are the cellular changes that are taking place during apoptosis and also necrosis? And also you need to know about the autophagy mechanism. That also play important topic, friends. So study about these topics followed by the protein sorting. Okay. So protein sorting, trafficking of protein to various cellular organelles, these topics are very important both with respect to CSI examination with regarding to BARC examination also it is very important. So last year in 2023 there was one question asked from signal sequences. Okay, which signal sequence is involved in importing proteins into nucleus, like that there were question has been asking. So please study those things clearly. And followed by the plasma membrane and functions of plasma membrane and membrane transport mechanisms. Okay, simple, active, passive, facilitated, pumps, various pumps. Last time there was question with asking regarding to channel protein, carrier protein. What is the difference between channel protein and carrier protein, sodium potassium pump or calcium pump, all those things. Next is enzyme inhibition. Okay, you need to study about all the enzyme inhibition, their graphs, an example of competitive inhibition, non-competitive inhibition, like that. Followed by that enzyme kinetics. So enzyme kinetic 90% the numerical questions like problem sums will be asking. So if you want to cover up biochemistry, you can cover up. If you don't want it, just leave the biochemistry completely and instead of biochemistry, study other units like molecular biology completely. Followed by that biochemical pathway. Gluconeogenesis, glycolysis, pentose pathway, Krebs cycle, and all those pathways with regarding to biochemistry. The most common pathway, and also you need to know about the location, what is the function, and also you need to know about the energetics, how many ATP are generated, how many NADPH are generated, like that you need to know. Next, you need to study about the structure of biomolecule, like if you are studying about carbohydrate, you need to know about epimer, anomer, all those things. With regarding to mostly they will be asking you need to study about the structure of DNA. Okay, they will be asking about the purine ring, pyrimidine ring, N carbon, how the N and carbon from which particular compound N and carbon are included, and also you need to study about nucleotide biosynthesis pathway is also been very important. Both the salvage pathway and de novo pathway you can expect questions from that. And then structure of protein. Okay, primary structure, secondary structure. While studying the structure of protein itself, you will be covering up the Ramachandran plot. At the same time, you need to study about NMR and X-ray crystallography. Okay, next is vitamins. So you need to study about all the vitamins because and also the function of those vitamins because many vitamins will be playing a role as cofactor. So you need to know about the vitamin, the function in which particular pathway that particular vitamin is acting as a cofactor. Next is Ramachandran plot. Followed by that, you need to study about the genome organization, nucleosome, histone, and different structural organization of chromosome. Like that, you need to know about all those things. So histone is again a very important thing. How histones are organized and about the nucleosomes. Okay, next is replication. Replication is again important. Replication, transcription, translation. You can expect from each topic. You can expect 2 questions. So when studying replication itself, you need to cover up DNA polymerases and other things. While studying transcription, you need to cover up RNA polymerases and their sensitivity to alpha mannitine, and also translation. You need to study about the tRNA, structure of tRNA, genetic code, all those things. Definitely you can expect questions from these topics. For sure you can expect a question. If you study molecular biology itself, you can score at least 30 marks out of 300 because 10 questions will be asking from molecular biology alone. Next, you need to cover up translational inhibitor, followed by the recombination are very important. The most important thing is DNA repair system. Last time 2 questions has came from this DNA repair system. You need to study both for prokaryotes and eukaryotes, and also in the DNA repair system you need to study about the type of DNA damages to DNA, and also most importantly you need to study about recombination repair system. Next is gene regulation. Again you need to concentrate more on prokaryote and phages instead of, if you can't able to cover up eukaryotic gene regulation, at least study about phage system and study about prokaryote like lac-operon, trip-operon, arabinosoperon, like that. Okay, so the next is cytoskeleton and cell signaling like GPCR and all those cell signaling pathway. You can expect 2 questions from cell signaling. Then cell to cell communication like cadherin, adherin junction, gap junction, tight junction, and then immunology you can expect many questions regarding to BARC examination, and also all the questions will be very easy for you to answer. So concentrate more on immunology also, immune cells and primary lymphoid organ, secondary lymphoid organ, and about the biology of B cell, T cell, and co-stimulatory molecule like that, and major histocompatibility complex. Next, autoimmune disorders types alone. There will be tabular column will be available in QB book. Just study the tabulated column alone, followed by that hypersensitivity reaction like type 1, type 2, type 3, type 4. Last time again one question it came from hypersensitivity also. Next is transplantation. Okay, so transplantation you can expect again 1 or 2 questions will be coming from transplantation. Next is antibody, types of antibody, and also they will be asking gel electrophoresis with regarding to antibody. Okay, so you need to know about how many bands you will be getting on digesting this IgA, IgB, IgM, like that you will be getting, and also about this pepsin digestion, papain digestion, that experiment, the popular experiment is there now. You need to cover all those things. Next is vaccines. Okay, give more thing about the corona vaccine. Okay, Covax and Covishield, how they had manufactured, whether it is an mRNA vaccine or whether it is a killed vaccine or whether it is a subunit vaccine. Next is complement system. Next is DNA marker. So you need to know about the type of DNA marker, which is codominant marker, which is dominant marker, and all those things with the gel pattern you need to know. Next is transposon, DNA transposon, RNA transposon, AC element, DC element, hybrid diseases, all those things. Next is ABC model of flower. Again, last year one question has came. So every year you can expect one question from this ABC model of flowering. Next is radioisotope. So radioisotope, definitely you can expect a question since it is a BARC which is related to this atomic and radiology field. They will be asking biologically important radioisotope because some radioisotopes are used for therapeutic side, some radioisotopes are used for diagnosis. Okay, next is photosynthesis pathway. Okay, in system physiology plant also there were many pathways starting from C2 photorespiration, C3, C4 CAM cycle, all the system you need to cover up. Okay, so just study about the tabulated column of all the photosynthetic pathways and photorespiratory pathways in plant. Okay, next is plant hormone. Next is pedigree chart with regarding to genetics. Regarding to microbiology, you need to know about the isolation of microorganisms, special focus to the microbiological media and factor affecting the microbiological growth. Okay, next is oxygen dissociation curve, left shift, right shift. While studying oxygen dissociation curve, you need to pay enough attention on high altitude physiology, deep sea physiology, all those things. Next is hemoglobin and myoglobin. So definitely you can expect 1 questions from hemoglobin and myoglobin. So difference between hemoglobin and myoglobin, Hill's plot and cooperation. There were many concepts will be coming under this hemoglobin and myoglobin. Okay, so study about, and also you need to know about the color of blood in different phyla. Okay, sometimes they will be asking what is the color of blood in this grasshopper or a sea squid, like that. They will be asking the color of blood in different organisms. Even in CSR examination also they had asked about the color of hemoglobin. Okay, next is bioinformatics. So bioinformatics also you can expect 2 to 3 questions. All the questions will be easy because last time I gave the BARC examination, and from that only I had learned these topics are very important. Okay, and also you need to study about molecular phylogeny, like maximum parsimony method. So phylogeny is also again an important thing. So study about the database types and all those things. Next is in evolution, you need to cover up a geological time scale like extinct organism, reason for extinction. Next is speciation, allopathic, parapatric, peripatric, and types of evolution like convergent evolution, divergent evolution, and also you need to know about the homologous organism, homologous organ and analogous organ. Next is fossil evidences, and they will be asking what are the list of living fossils. Okay, next is evolution theory. Darwinian theory, Lamarck theory, and what is neo-Lamarckism, neo-Darwinism, like that. So evolution just cover up basics. So if you ask me, just cover up whatever given in the Pranav Kumar sir book. That's well sufficient for you to cover up evolution. Next is blotting. Okay, northern blot, southern blot, western blot technique, all those things. Next, isolation of DNA. What are the chemical? What is the significance of those chemicals that are involved in isolating DNA, RNA, and all those things? Okay. So next is about the techniques like CD spectroscopy, NMR, X-ray. So CD last time one question was asked from this CD spectroscopy. Okay, so CD, NMR, X-ray are very important. Then PCR, basics of PCR and about the list of enzyme. Last time there were one question asked from tag polymerase. Okay, and also types of PCR with regarding to real-time PCR, they will be asking the dye. So in which part of DNA the cybergene will be attaching. So those things you need to know, friends. Next is sequencing. Okay, next generation sequencing, up to next generation sequencing you need to know. Next, in developmental biology only very little bit questions will be asking from developmental biology. So just try to understand about the fertilization. You need to know about the features of egg and sperm. Next is restriction enzyme digestion regarding to RNA technology. Next is genetic diseases like chromosomal diseases, single gene diseases, like that. Next is mutation, type of mutagens and their effect. And while studying mutation itself, you need to study about Ames test for mutation, which organism is employed in Ames test. Next is microscopy, types of microscopy and all those things, and immune techniques. This include ELISA technique, agglutination, precipitation, radio immunoassay, ELISpot, all those things. Followed by that, disease, human disease caused by bacteria, virus, fungus, protozoan, like that. Next is viruses. So viruses definitely you can expect, you need to know about the genome of virus and the route of entry of the virus, like that. Next is gene knockout technology, which include Cree, LogSP recombination. Definitely you need to study about gene knockout, which is very important technique, friends. Next, ecology and all, very few questions will be coming from ecology. If you want to skip, you can skip, but my personal suggestion is that just cover up the important topics or the topics which you find it is easy for you. Okay, next is transgenics, complete transgenic. So list of transgenic animal, list of transgenic crops. Sometimes they will be asking which is the first crop that was approved, transgenic crop that was approved in India, like that they will be asking. Recently India has announced a transgenic mustard, like that. So you study about the transgenic mustard which is cultivated in India. Don't go for overall world, just cover up the transgenics animal and crops in India. Next is protein charge calculation, amino acid. So sometimes they will be asking you about the amino acid. So hydrophobic amino acid, polar amino acid, there were structures and all, they will be, you need to follow the structure, which amino acid will be having a benzene ring, like that they will be asking. Next is chromatography, types of chromatography and all those things. So bioinformatics I have previously had covered up. Next is plant breeding, special reference to marker assisted selection. Plant breeding is very important. Last time 2 questions has been asked from plant breeding, so please study them completely. Next is peptide digestion, exopeptidase, endopeptidase, carboxypeptidase, aminopeptidase, where they are cutting, whether it is in N terminal or whether it is C terminal. Next is sterilization methods in microbiology. Next is antibiotic, antibacterial drugs, antiviral drugs, and antifungal drugs. Next is bacterial genetics, transformation, transduction, and conjugation, all those things. F plasmid, HFR plasmid, what is the difference between F and F dash plasmid, all those things. Then next is genetic term, common genetic term. Mendelian principle, what is mean by pseudo dominance, what is phenocopy, what is maternal inheritance, what is maternal effect. So basic genetics term just find out. Next is gene mapping method, and also you need to study about post transcriptional modification. Like last time 2 questions has came from this particular thing. Okay, so you need to know about splicing, capping, polyadenylation, all those things, and also you need to study about decay, mRNA decay, and all those things. So post-transcription modification very important. Even though if you can't able to cover up the transcription completely, just cover up the post-transcriptional modification. What is happening after transcription? How mRNA are capped? What are the enzymes that are involved in polyadenylation? How introns are removed, like that. Next is post-translation modification and hybridoma technology. Flow cytometry is again a very important thing. Flow cytometry followed by fluorescent activated cell sorting are very important. Next is types of DNA, A DNA, B DNA, Z DNA, and types of RNA. Next is neurotransmitter. So types of neurotransmitter, their biosynthetic pathway or precursor, and also while studying neurotransmitter, you need to study about action potential also. Next is chromosomal banding techniques. Next is cell cycle, and their regulation is very important. So both mitosis and meiosis you need to know. They will be asking how many chromosome will be there in this phase, that phase, and also they will be asking which is the smallest phase in cell cycle, like that. Those questions you can expect, which is very basic level. Only thing is that you need to study, you need to at least revise once all those things. Okay, next is gene regulation, antibody engineering like allotype, isotype, autotype, and recent antibody engineering you need to know. Next is immune response to infectious diseases. Infectious diseases in the sense viral diseases, HIV, malaria, common diseases. Next is sensory photobiology. This is with regarding to phytochrome, cryptochrome in regarding to system physiology plant. Okay, next is secondary metabolite. Next is blood group. Of course, blood group is very important. A group, B group, and this O blood group, all those things. Next is endocrinology, the hormones and their function. So next is chromosomal aberration. As I mentioned you earlier, chromosomal aberrations are very important. They will be asking which chromosome is affected, Robertsonian translocation, and all those things, inversion and deletion, biosensor. Recently in CSIR, December 2024, there was one question asked for biosensor in 4 mark. Vectors again a very important thing. Then biostatistics. Biostatistics are just very simple. Mean, mode, median, and skewness and central tendency, like that. Next is mice, kid mice, nude mice, gene therapy, and human genome project. Recently there were various innovations was made on human genome project. Just try to understand what is going on in human genome project, what are the recent developments in human genome project, all those things. Next is biodiversity conservation followed by types of RNA, starting from your rRNA, mRNA, siRNA, miRNA, and all those things, how they are produced, what is the precursor of miRNA, what is the precursor of siRNA. Next is blood clotting. You need to study about the pathway of blood clotting and what are the list of blood clotting drugs, and there were different factors were there. They will be asking you the name of factor. Okay, so these are the important 100 important topics regarding to BARC Biosciences examination. If you cover up these 100 topics, definitely you can score 50% of your mark, or you can score the mark that above the cutoff. Okay, so in the next video, maybe I'll be completed telling you the syllabus for BARC examination. Okay, so if you want some other help, please do tell me. Thank you, friends. Thanks for watching this video.